Thank you, Vicky. Good morning, everybody. Great to see you. Fortunately, it's not snowing, but in some ways I wish it was, because it'd be cold enough to suppress some of the aphids that we may have to deal with later in the season. So what I'd like to do is build on what you've already heard of this morning and maintain the theme of integrated pest management in this particular case and concentrate on virus yellows, which is basically one of our biggest threats to the UK crop. And just reflect on what we do, because in many respects we are already adopting many of those principles in how we go about controlling this quite difficult pest that causes many challenges to you all. So when it comes to virus yellows, we have been using since the 60s the virus yellows forecasts and modelling and prediction systems that are really the envy of Europe. And when we talk with colleagues in Europe, they're now trying to catch up with some of the strategies and tools that we have to be able to monitor and predict when the first Mysis persicii, for example, may land in a sugar beet crop. And I think that's really important just to emphasise we do have these tools. A photograph from the article that's in the Beet Review written by the scientific team at Rothamsted, where we are taking first flight information. We are using that to predict when they're first going to arrive, taking into consideration the crop growth stage, which then is influential of when you may or may not trigger the use of a foliar insecticide. And these are really powerful tools that you are using day in, day out, and probably take for granted. We also have the very powerful information about, and we're right in the middle of that critical period at the moment, about the influence of January and February temperatures on aphid buildup and when they may arrive. The data in front of you is going back to 1962, and the milder that condition is, the earlier those aphids will migrate. And just listening to the news this morning when we're talking about we've breached the 1.5 degrees temperature threshold, the consequence of that, when it comes to Mysis persicii, means they're probably going to be in your crop at least 12 to 15 days earlier than they would have been by having that time influence. So it's really important we take this, because it does mean then those aphids are coming in earlier into your crop when they're at a smaller growth stage, and we need those interventions to be able to minimise that. All these tools and all this information has been really critical, and it links through to what Hazel's just said about the use of all the necessary control strategies for limiting particularly aphids and virus yellows. We all know the history behind the use of the neonicotinoid seed treatments. They were first introduced in 1994 into the UK and were withdrawn in 2018. But clearly, as an industry, we need solutions that you're going to hear about quite eloquently this morning from some of my colleagues about trying to find those alternative IPM solutions. But the seed treatments have provided that protection, particularly in the short term, to ensure that we can protect your crop, keep those aphids out, and minimise the impact of virus yellows. But when we have gone for a derogation, using those IPM principles has been key to DEFRA to make sure that we can actually gain a positive response, to ensure then when we get to a trigger point, you can actually deploy it. And clearly you've just ordered your seed, and some of you will have made that decision about using the neonicotinoid, i.e. Cruiser SB. In 2020, that wasn't an option, and clearly many of you will remember the consequence of that season and the widespread impact of virus yellows after an incredibly mild winter, which led to 38% of the crop going yellow nationally, over £60 million lost, and for some of you, 100% of the crop did go yellow. In 2021, 2022, and also in 2023, we applied for derogations using the forecast and predictions. And clearly in 2021, when we did have the beast from the east and a very cold period, the threshold that was put in place actually wasn't breached, so we didn't use the seed treatments. In 2022, DEFRA reappraised the threshold, increased it slightly, but we had that very mild winter, and in 2022 we had the situation where we were able to trigger the use of those seed treatments. Sadly, 2022 was quite a difficult year for lots of other reasons, but clearly you've got to remember the threshold trigger point is in the absence of any other mitigation that we may need to put in place, which then you will bring into the crop as we see the threat develop. So what happened in 2023? Well, if you remember, we also applied for the emergency authorization. DEFRA increased the threshold into over 60%. But because of the very mild conditions, and you can see from the Met Office chart on my right, you can see we had an incredibly mild winter once again, and the entire area was at threat. So consequently, it meant that the use of cruiser was available, and 60% of the area was actually protected. I always find that the first predicted aphid migration is a really important part of what we do and we watch very closely within BBRO. But it's so powerful because it's often only out by hours. You know, you're predicting when the first aphids are going to be coming into your crop back on the 1st of March within about 24 to 48 hours. That's incredibly powerful and enables us to then predict all sorts of strategies, and clearly we communicate that in the bulletin to whether or not you have used cruiser about the strategies. And all this long-term data is really quite important then for how we go forward through the crop, because if you look at the 2023 season, actually some of you will have not needed to put in any further intervention, because many of you will remember, particularly if you're in East Anglia, for example, we actually had a very cold spell, northerly drift. You can see most of the Cantley area is blue for that particular period. And although the crop continued to grow, this suppressed aphid activity and reduced the pressure potentially of virus spread. And what you can see from the chart is BBRO, with colleagues from British Sugar and agronomists, were monitoring at 46 sites with and without neonicotinoids. And what that clearly demonstrated was the value, once again, of the seed treatments at minimising the risk of aphid buildup. But also then, if you hadn't used a seed treatment, this information was really important to get into check your crop and look for the one green wingless aphid before plants to put in any further foliar aphicide sprays. So when it came to virus yellows, the good news was for 2023, although the risk was high, within season variation and consequence, along with all the chemical and other approaches and good hygiene and mitigation you deployed, ensured that virus yellows was relatively low level for that crop and really had little consequence on yield impact. But what I'd like to do now is hand over to Alistair Wright, my colleague within BBRO, to bring you right up to speed with various initiatives that you've been probably keeping a very close eye on, the work on resistant varieties and other mitigation. And Alistair will bring you right up to speed before he gives the opportunity for Sasha to say some words with regard to a new review BBRO are undertaking to look at spray thresholds. So I'll hand over to Alistair and he will guide you through those findings.
Good morning, everybody. It's a pleasure to be able to stand up here and, as Mark said, give you the update on the vast array of IPM work BBRO is undertaking to find these alternative solutions. So I don't make any apologies. You are going to see the IPM triangle a lot, as it does underpin quite nicely the approach we are taking and highlights the whole range of work which should deliver to you cleaner, hopefully virus-reduced fields. But of course, it's not just virus yellows, and these principles need to be applied across the board. But I think it was nicely highlighted earlier in one of the questions, isn't Regen Ag and I suppose IPM as an extension of that just good farming? Yes, of course, we all want to be out there producing high-yielding crops, but we are having to look for these alternative solutions. On that previous slide, I showed a whole range of pests and diseases, but we will be using virus yellows as our model for what I'm going to be talking through, but these principles can be applied elsewhere. But we know virus yellows very much remains the greatest threat to our crops, not just here in the UK, but across Northwest Europe as well. As a quick reminder, for anyone who needs it, we are trying to deal with 3 different viruses simultaneously. So we are looking at beet chlorosis virus, which causes at most up to around 25% yield loss. That's the one here on the left. In the middle we have beet mild, which is the sister virus to beet chlorosis and causes up to 30% yield loss. But our greatest threat remains beet yellows virus. causing up to 50% yield loss, very rapidly transmitted by the aphids as well into the crop. So that is the one that is the Achilles heel, and we must look at all these alternatives in a post-neonic situation to managing virus yellows. And of course, I don't think they need any introduction. Public enemy number one remains Mysis persicae here, especially the winged form, that, as Mark has said, we can now very accurately predict when they'll be arriving. But these are what will start transmitting virus into your crops and also producing their progeny, which then go on and colonize patches in the field. I'm sure for many of you, you don't want to think back to 2020, so I won't drag you back quite that far, but just to highlight here, this was a field at Brooms Barn that I'm sure many of you are familiar with as the former research station for the crop, but the field here is almost completely infected with beet yellows. This was in the absence of any control measures, because this is for experimental purposes, but you can just see how widespread, even in a year when we weren't predicting a massive epidemic, without any control measures, the crop goes very much yellow. IPM, however, I don't think will deliver completely clean, virus-free crops. We've got to learn to accept a certain level. This is one of my very own sugar beet fields near Thetford, and you can see the patches of yellow through the field, and probably a few more than I'd have liked, but this is the kind of vision we need to be heading for. Isolated patches. I don't think we're at the stage where we can just be limiting virus infection to individual plants like the neonics delivered, but through the use of all the other tools, hopefully we can very much reduce the spread of secondary infection. So how are we going to do that? We'll start at the bottom, good on-farm practice, which should build those solid foundations. Of course, I imagine and I hope many of you are doing some of these things already, but some of them are probably a bit more nuanced and require a bit more refinement, but we are here and we are undertaking research the whole way to deliver these solutions. Now, my first message is one that you can definitely address when you get home this afternoon, although maybe not when it's this wet, but is to go and clean up any spoil heaps from this year. The spoil heap on the left was left, I think, behind a mouse, and you can see lots of large root fragments there, and when they start to sprout in the spring, they're a perfect place for the aphids to emerge from. And on the right here is a very special aphid 2 BBRO. That is the single aphid that delivered our beet chlorosis culture and has now gone on to infect several 100,000 plants, all from one aphid. Of course, in the field, we don't think it would be quite that drastic, but it just shows you the potential that these have. So definitely good hygiene is critical. The next step, and hopefully one that we'll see continue to develop well into the future, is the use of tolerant varieties. And this will be a crucial foundation for your IPM to deliver effectively. I'm sure many of you, or all of you, I hope, are familiar with the fact we have one on the recommended list already. KWS's Marussia. And it's very characteristic for this very dark canopy. And it stands out well in trials and in fields where it's grown in strips. But as you can see from the recommended list, unfortunately there is that expected yield drag that comes from introducing new traits within Marussia, and it only is claiming partial tolerance to beet mild. So we need varieties that are going to perform well under all the viruses, but that also in the absence of virus are yielding way up this end of the RL, because of course losing 10% yield before you've even got virus in the field can be a problem. So we continue to operate our vast virus yellows testing program, and it rolls forward for another 3 years of Goliath. So we've been running this since 2019, looking at new novel varieties and novel sources of resistance from the breeders, and I thank them all for their offerings, because without that there's nothing to test. And these varieties, you can see the beet yellows plots here, still standing out quite clearly. But the most exciting step forward is there's now stripes in there. They're not going all completely yellow, and that is showing itself in enhanced yield performance as well. So I'm going to take you through this graph now, and I don't apologise that it's going to take me a little while, because it's a really important message about how far these variety types are coming. So I've plotted across the bottom here the percentage of beet yellows infection. So this is the most severe virus. So the 100% being the fully inoculated plots, very worst case situation, 0% being the controls. And you can see here the first variety we plot on is BTS1915. And we know that's flying high at the top of the list, yielding fantastically well. And we've seen for many years now, it sort of has this 35% or so yield impact. So not as high as that 50 that we could well expect under beet yellows, but of course not acceptable still to let a whole field go this way. But it does do better than this variety, which is Daphne. And Daphne is good for trialling because it shows some nice strong symptoms, but you can see it loses a little more to beet yellows. And then we have a more traditional variety, lacewing here, that's losing well over 50% of yield, and that's not a surprise. None of these varieties so far claim any tolerance to the viruses. And again, lacewing is another good indicator. But now we put Marussia on the map. And as I said earlier, it only claims that partial tolerance to beet mild, but the line here is much shallower, less steep, and you can see where they start to intercept is when this variety starts to outperform the others. So of course 1915 still is about the same as Marussia, and you've lost this at the lower infection levels, but if you've got Daphne and lacewing or those traditional variety types, you could consider using these new varieties. Most excitedly now though, we plot in some of the new novel material that is coming through. And you can see here there's a massive uplift in the yield potential, and this line has the same sort of response as Marussia, and we're losing around 25%. So remember, this is beet yellows, we'd expect 50, we're losing 25 instead. So that's from one breeder, and it is the same story from another. So we're seeing progress not just from one breeding house, but from several. Actually, in fact, all the breeders that are partaking in the trial, we're seeing good improvements year on year. So when we just simplify that, you can see the uplift is consistent, and this is fantastic news for everybody. So there is significant progress. They are definitely on the horizon now and really coming into focus, these varieties, and this next generation I think will start to be a game changer, and they're expected to be on farm as early as 2026. But the most important thing is they are well up amongst the pack with the RL in the absence of virus. So that's variety change, and that obviously is a decision you need to be making in the autumn. You're not going to know what the virus pressure is going to be. So if you have still selected the more susceptible varieties, and we won't see a direct shift overnight from the traditional varieties to VY varieties, so we do still need a range of other options. And we believe companion cropping has many benefits, and we have been working in this area now for several years. And they can broadly be put into several categories, but there's a great deal of overlap between them. In 2020, we helped monitor several sites that had used barley companion crops, mainly to stop windblow. And this picture here shows an example, again, on my own farm back in Norfolk. And we did this in 2022. And the beet without the barley, of course, were nice and big, protected from the windblow. The ones that were grown amongst the barley were definitely held back by the competition. And we believe there's definitely a benefit for virus control from using these companion crops, but you need to be spraying them off before they become far too much of competition for resources. And just a good reminder there is the yield data we obtained in 2020. And in most sites, there was a reduction in yield when the barley was used. So although agronomically or visually they looked better, there was a severe impact. So the way to reduce that obviously is to kill that barley as soon as it's sort of reached cover. It'll be masking the sugar beet, but you want to then stop it competing for water and nutrients. Now the clicker's working again. We're looking at attractants and repellents as well, and we've been using these as part of our variety testing for a long time now. We always flank the inoculated plots with rye. That acts as a natural windbreak, stops the aphids, the hundreds of thousands of aphids we're putting out in these fields from being blown away. But if they do get over the rye, there's a brassica there to hold them. And the brassica would be a preferential host. The aphids shouldn't want to wander too far from there. And there's the benefit there of those brassicas not hosting the sugar beet viruses. So this could be an option, and this spring we should be having some work looking at putting some brassica strips through some commercial fields and see how that pulls aphids away. But of course we need to wait for the aphid pressure to be significant to be able to do that. Other options, and one that's showing some real promise, is the use of these endophyte grasses that have been bred in New Zealand. And this is just a snapshot from the drone of a field at Morley near Wymondham in Norfolk, where we had the endophyte grass sown as a companion crop here. And between here it was sprayed off, and there's controls running either side. And the beet definitely stayed greener, and in this previous photo you can see where the endophyte strip was quite clearly. But again, there's the issue of intercrop competition going on. But I'm delighted, and Athrea's here today. BBRO and the Morley Agricultural Foundation are supporting a one-year MRES project at the University of Nottingham, really to look at the agronomic impact of these endophyte grasses and what benefits they could bring. Using wildflowers is also definitely in the news at the moment, and even on the television, with McCain's flouting it as a crucial part of their regen ag strategy. And by all means, they should be used for sugar beet as well. We have listed these species, and they're in this flower strip here, again, which was at Morley, and was designed to host the beneficials by providing a pollen and nectar source. And by all means, they look fantastic, and they were buzzing and full of bees when I went to look at them, but they weren't delivering the resources, the pollen, that nectar, early enough to host the beneficials when we need them to then go out and scavenge for aphids. So I think they need to be part of a ecosystem-based service. They become a permanent feature on farm because we're not seeing the up to 100-metre control from the strips that the literature claims there should be. And there's been a lot of work done on these across Europe. But by all means, I think they need to look like this. In the summer beforehand, you need to have a vast array of different species to host the beneficials, give them some overwinter habitat as well. And thankfully now with things such as the SFI, there should be some fairly rewarding cash incentives to keep them as a permanent feature on farm. So that's sort of those solid foundations we need. Of course, that's only going to take you so far in controlling the aphids, and Mark has touched on the forecasting and monitoring work we continue to undertake. But you need to remain alert. And you can see that there's a comprehensive article in Beet Review which takes you through all the modelling, so I won't dwell on it. But we know this winter temperature around now, up until the end of the month, really drives aphid abundance. And there's some real solid science behind that. The reassuring thing is we're now looking at how we can regionalise our model. At the moment, it only is based off the 2 suction traps in the sugar beet growing area. But we are now funding a new project to look at regionalising or even localising that to a postcode level. It'll depend on what the model can produce. And that's being done in collaboration with Rothamsted Research and James Bell, who's now moved to the University of Kiel. But of course, we continue to offer our monitoring service in-house too. And Mark and the rest of the crop protection team will be busy fishing aphids out all spring. And we'll have yellow water pans out at around 17 sites this spring, and around another 30 sites that will be monitored for aphids in the crop. So you'll be able to log onto the BBRO website and check your local hotspots, and that should start giving you that early warning to then go out and check your crops. Of course, when you find aphids, what can you do? Or more importantly, when we predict that you're going to find aphids, what can you do? And that's probably when we need to look at these alternative strategies that can either prevent the aphids from even landing in the fields in the first place, or confuse them. So that is when these other interventions can come into play, before we have to then resort to chemical control. We've been looking at on-field dyes and changing the colour of the soil now for several years, and I was very sceptical to start with, but there is some solid science behind this, and it's all about changing the reflectance of the soil, and that is how an aphid mainly finds its hosts. And it gained a lot of traction through the BBC, and I had interest all the way from Canada and Florida as well. And Anna Hill was taken by it for Farming Today. But I thought, yes, this probably won't work. But we've seen some really exciting progress this spring from one of our sites at Rougham. And you can see there's a significant decline in the number of aphids found in the dyed plots. And it didn't matter actually which colour of dye. They all had an effect in reducing the aphid number. So that's positive. The downside is at this stage they were all above threshold still. So earlier on though, and especially stopping those early aphids that we know are the ones that mainly carry the sugar beet viruses into your crops, this could be a really viable strategy. We're now needing to look at how we keep the colour as vibrant for as long as possible before it gets washed away or fades, to provide as much benefit. The other good thing, and I was worried there would be a phytotox issue, but no, there was no significant reduction in yield when they were sprayed with the dye either. So that is a really good, promising piece of IPM. We also are looking at deterrence. So instead of killing the aphids when they land, make sure that they don't land in the first place. And we're now partnering with a French startup called AgriOda, who have been funded in part by the PNRI, which is the French national initiative, which has had 12 million euros of funding from their government to look for solutions for virus yellows. For anyone not fluent in French like myself, I'll just point you through some of the main highlights, but we will be generating some UK-based data this year. By putting these beads down that are embedded with a proprietary odour mixture that AgriOda own, there's up to a 10 to 15 day delay in reaching threshold. That is really good news because that allows your beneficials another 2 weeks to come in, lay some eggs, they can hatch, and they can start controlling the few aphids that have landed. And that delay then affords the beet also some more time to grow. So really interesting, positive steps. And these can be simply spread on with a slug pelleter. So we think this could be a really exciting option for deterring the aphids. And by using our thresholds and also the monitoring when we expect the aphids to first appear, can then be used as a trigger for applying the AgriOda. Beneficial insects, I've touched on this already with the flower strips, but we don't really recommend anyone goes and buys some commercially. They're hugely expensive and they're quite soft. When they're raised in a glass house, they don't quite have that ability to cope with the big diurnal ranges in temperature. So the best approach is definitely to host them on your farm over winter. And by that point, the likes of the lacewing larvae and the ladybirds and the parasitoid wasps will then be ready and primed for when the aphids may appear. You've still got to go and inspect your crops though, of course, and if we do need to look at making some other interventions, we need to be using the threshold. Mark here, you can see, is out checking these plants, And I'm sure we've said it several times this morning, but that threshold of 1 wingless aphid per 4 plants still stands, and that is really where we're investing some money to review that. What's going on here? Right. So if you do reach threshold, of course, there's the need for chemical intervention. So hopefully everything you've done beforehand can help prevent that threshold or the need for multiple aphicides, but of course, in years of high pressure, you may need to get the sprayer out. I won't reiterate it again, but 1 aphid per 4 plants, and at that point, you then need to make a decision with your agronomist on which active ingredient you may wish to use. These are, of course, the foliar options. So you've got Topeki or Afinto, with the active ingredient being flunicamide, or you can use Insist, acetamiprid. So they both have their benefits and drawbacks, but of course, timely application and controlling the aphids as early as possible is very important. So I'll now pass on to Dr. Sasha White, who has provided us with a video because he can't be with us today. But we at BBRO have appointed ADAS to undertake an independent review of the spray thresholds and a few other options around aphid control. And that work Sasha will now present.
I'm sorry I can't be there today, but hopefully this video will suffice for now. I'm going to be giving a short talk on a new project that's just started. It's called Reviewing Models and Data for Use in a Virus Yellow's Decision Support System. We do have a little bit of support in knowing when to use them, so we've got some thresholds. But there is, a question about how robust these thresholds are. What is the basis of them? And are there grounds for updating them? And also, if we are going to update them, should we make them more dynamic? So should we make them shift, go up or down, depending on the condition of the crop at the time? So for instance, they could take account of variables such as the effect of weather on the population growth of the aphids, the proportion of the aphids that are carrying the virus, crop economics, all of these things and others could be important. And if we're going to do that, then the best way of doing that is through a decision support system or a model that helps the user make those informed choices. So the project's going to be carrying out a literature review, looking at the basis of the treatment thresholds, seeing whether there are other thresholds and whether there's any grounds for updating them. It's also going to be looking at whether there are any models out there on epidemiology of the viruses, the population dynamics of Mysis persky in crops and their movement between plants, and also sugar beet growth models. And where any of these models aren't available, we're going to be looking to see whether there are useful parameter values that we can use in building our own model. And when I say parameter values, these are values that are important for the system. So for instance, the rate at which a plant gets infected by an aphid, and maybe the effect of temperature on the reproduction rate of the aphid, those sorts of things. Ultimately, we'll be providing some recommendations on the structure of a possible future decision support system that can help guide sprays for virus yellows control, and also identify where there are any knowledge gaps, so where we might need to generate our own data for building a decision support system. Now we have a little bit of track record here. We developed a BYDV decision support system for winter cereals recently. It's called the ADAS Crop BYDV Assessment Tool, or Acrobat for short. And this is a schematic of it here. Essentially, it's made of 3 submodels. So the first is an aphid population model, simulates the aphids in the crop, their population dynamics. That feeds into disease epidemiology submodel. That simulates the infection of the crop. Then that feeds into a risk model. And essentially what this does is it takes the simulated or the predicted number or proportion of the crop that's infected. It calculates what the likely yield loss is going to be, and then makes a cost benefit analysis of whether a treatment is economically worthwhile at that point. On the left, you can see all the input factors and variables. So the numbers of alate aphids flying, weather data, things like sow date, the proportion of aphids that are carrying the virus, and crop economics. And then on the right, you can see the output. So a risk level and a spray recommendation. So you don't need to worry about spraying, or you might want to consider spraying. Now for Acrobat, it was field specific. It took account of all sorts of really important variables, and we validated it in field trials across 2 years. And in those field trials, we found it accurately predicted infection levels, and also provided effective control, i.e., recommended sprays where they needed to be. And this graph here just illustrates the sort of things that we can do with the model. So here we can look at the effect of sodate, for instance, on BYDV infection. So we've got 3 different crops here. So we've got an early sown crop on 1st of September, that's the blue line, a mid-sown September, or late September sown date, that's the red line, and then a late sowing date, that's the end of October, that's the green line that's tracking along the bottom. And you can see the effect that these different sow dates are having on the infection rate in the crop. So the early sown crop gets infected much more quickly than the late sown crops. And you can also see where the sprays were being recommended in these simulations. So in terms of progress, well, we've only just started the work a couple of weeks ago. We've started looking at the thresholds. They're based on work in the 80s, so certainly potential grounds for updating those. The way that we grow crops has changed a lot since then. There does seem to be very little other work done on looking at thresholds, but there's much more to do. So hopefully I'll be back reporting to you next year. And just to thank everyone who's been involved in the 2 projects that I've mentioned here, and also all the funders. And thank you for listening.
So Sasha's not here today, but if anyone has any questions about that project, Mark or I will take them later. But it is a really exciting opportunity to reflect on the threshold and then look at the viability of developing a new decision support tool. But in the meantime, of course, we need to keep going with the IPM options we have. But the most exciting thing is that it does look like there are new solutions coming and on the horizon, especially for managing virus yellows. But it does need that integrated, multi-pronged approach. Decisions will need to be made on a field-by-field basis, and that will take more time. But that attention to detail will be key, but hopefully it will deliver back to you in enhanced yield. And by using the IPM triangle as a guide, but by no means a blueprint, it should help you protect your crop and the local environment. And hopefully I've reassured you that we as BBRO continue to make significant investments in the IPM area, and we will look at delivering durable and viable IPM options back to you.